Give us 1 minute.
Welcome to today's Open Research webinar hosted by eLife. This series aims to give early career researchers an online platform to continue to share their research as an alternative to in-person gathering. I'm Anna Akhmanova. I'm a deputy editor at eLife, and my own research is focused on the cell biology of the mammalian cytoskeleton. It's an exciting moment for me. It's the first time I'm chairing a webinar, and I think it has a very important function to connect people in this very difficult time when we're all confined to our homes and trying to make the best out of it. And we are particularly excited here today at eLife to be able to give some of our young researchers who cannot travel to meetings anymore an opportunity to present their research and to connect to many of you. Today we will hear talks from Sejal Davla, who is a research associate at Advanced Science Research Center, City University of New York in the United States. James Thomas, postdoctoral fellow at Fred Hutchinson's Cancer Research Center, also in the United States, and Ryoji Amamoto is a postdoctoral fellow at the Harvard Medical School, also in the US. Okay, so before we start with the talks, just a few explanations on how we will going to proceed. So after each talk, which will take 10 minutes, we will have 5 minutes for questions for the speaker. Then we will move to the next talk. To ask a question, you can type it into the chat on Zoom or directly in the Google document linked here. The link is also present in the chat. We are joined today by Miranda, Anya, and Naomi from eLife, who are working in the background to support you. They will help me line up your questions. I will read your questions out loud and include your name where possible. The open notes document is also a place for you to continue shared public notes. We welcome you to do so and to list yourself as a contributor in the list above, the speakers for today's webinar. Thank you. Finally, I'd just like to let you know that we are also recording the webinar and live streaming it on YouTube. During this live webinar, we ask you please be respectful, honest, inclusive, accommodating, appreciative, and open to learning from everyone else. Do not attack, demean, disrupt, harass, or threaten others, or encourage such behavior. If you feel uncomfortable or unwelcome on any of these webinars, please contact eLife by email. We have an inbox which is watched by Miranda and I at eLife. The eLife organizers reserve the right to ask anyone to leave and to deny access on the subsequent webinars on Zoom. If you need any help at any point, please send Miranda, Naomi, or Anya a chat message directly using Zoom. Okay, so we will now start with our first speaker, who is Sejal, and she will tell us all about it. The title is ANT1, Functions in Astrocytes to Regulate Sleep Homeostasis. Sejal, welcome to the webinar.
Thank you, Anna. So I begin to share my screen. Just let me know if it works. We good? Can everyone see my screen? Hello.
Hi, Sejal. It's Naomi.
Hi.
Hi.
Do you hear me? Did you see my screen?
Yeah. So we can't see your screen right now. If you try exiting and share screen again.
Does it work?
Not yet.
Huh. Because I see it here. I did the share screen.
If you close presentation, are you in normal PowerPoint?
Yes.
Yeah, so you're not presenting right now. It's your PowerPoint window.
How about now? How about now?
No. Let's work out how to do this in the background. Perhaps we should we go to James? James, would you be ready to go first? And then we'll come back to you, Sejal. And in the background, we'll work this out.
Yeah, I can present. That's fine.
Okay, excellent. If we can do that, let's do that. Sejal,
yeah, absolutely.
Okay, then we will first start with James Thomas. James, welcome, and please share your screen.
Great. Can people see my screen here? Hold on, I'm trying to see.
Yeah, I see your screen.
Great. Sorry, I was pulling up the chat to make sure I could see it. Okay, great. Yeah, thank you so much, everyone, for organizing this and having. Let's start my video. Okay, perfect. So everybody's good. Okay, let me know if you can't see anything. Great. So yeah, thank you everyone for having us and yeah, like participating in this during this difficult time. I hope everyone is doing well. Okay, so my name is James. I'm a postdoc at the Fred Hutchinson Cancer Research Center, and I'm going to talk to you today about RNA splicing and new tools that we've been developing to study RNA splicing. So here I'm showing you a very simplified gene model. So there's an exon here that has a start codon, and an exon downstream, which has a translation stop codon. And these two coding sequences are separated by an intron. And during the process of RNA splicing, the introns are removed, and you can kind of think of the introns as quote unquote junk. So this junk gets removed. Now the exons are ligated together. And now this coding sequence can be translated to make protein. So you can sort of think of the coding regions as being the quote unquote important parts of the message. And the importance of these coding regions are consistent with the conservation we see. So here I'm showing you these blue regions here are regions of high sequence conservation. And if you just take a quick glance, it looks like the intron really has no conservation whatsoever or upstream, but really I've been hiding these regions of incredibly high sequence conservation across this gene. And these are actually regions known as ultra-conserved DNA elements, which are defined as regions greater than 200 base pairs in length that are perfectly conserved between human, mouse, and rat, and also very highly conserved in even more distant organisms, such as the pufferfish. Surprisingly, most ultra-conserved elements are in non-coding regions of the genome, and they're often located within or adjacent to essential genes. So DMRT3 is a gene that's known to be important for viability in mice. So if you knock out this gene, there's phenotypes you see in a mouse model. So you might think, great, let's delete one of these ultra-conserved elements. Since they have all these remarkable features, certainly the animals would have viability defects. But when this was done, using a single ultra-conserved element knockout, the mice were viable, and there was no postnatal phenotypes. So this was very surprising. It turns out actually more recent studies have shown that combinatorial deletion between two ultra-conserved elements actually begin to unmask some phenotypes. And this suggests that these ultra-conserved elements could be functionally redundant. So here I'm going to show you another example of some ultra-conserved elements in other surprising regions of the genome. So here I'm showing you the gene structure for SRSF3, which is an RNA binding protein. And again, the coding sequences are very highly conserved, but again, I'm hiding something, and that's this other exon that has incredible sequence conservation across it, but also very high conservation within the flanking introns. One other thing you might notice about this exon, which might be a little surprising, is it contains a premature termination codon. And we call this class of exons poison exons. And we call them poison exons because when an exon gets spliced in and introduces a premature termination codon, this is a signal for that transcript to be degraded by a process called nonsense-mediated decay. So essentially, poison exons poison the host transcript. So you get the exon in, and the transcript gets degraded. These poison exons tend to be enriched in many RNA splicing factors, such as SRSF3, and they are speculated to provide, speculated but also some direct evidence showing that these provide a mechanism of autoregulatory feedback. For example, SRSF3 encodes an RNA binding protein, which can regulate the inclusion of its own poison exon. So this sort of provides the cell a feedback loop to maintain the levels of SRSF3 and other RNA binding proteins. A lot of these poison exons, we can actually see them in even more distant organisms, such as certain molds. Again, really highlighting the, the high conservation of these elements. So again, intriguing and almost obvious hypothesis is that these poison exons must be important for organism viability. The challenge, though, with studying poison exons and really splicing in general is that there aren't, until we hope recently, great tools to study these, especially in a high throughput manner. So again, this is an isoform that would contain a poison exon, and this would be degraded by nonsense-mediated decay. And the isoform on the right would make a functional protein. So we want to ask the question, what happens when we just delete this isoform? Unfortunately, we can't use RNAi to do this. You could envision designing an siRNA to target the poison exon, but that just copies what the poison exon does in the first place, which is knock down the message. And really, we want the message to go up. Another strategy would be to use an antisense oligonucleotide to block RNA binding proteins that regulate the inclusion of that poison exon. But ASOs are low throughput, and it can often be challenging to predict which ones will be effective at blocking a certain exon's inclusion. So we decided to take advantage of CRISPR-Cas9 to delete these poison exons, and more specifically, use pairs of guide RNAs in a Cas9 expressing cell to delete the exon. And this was inspired by this paper and others showing that delivery of pairs of guide RNAs can lead to deletion of large genomic segments. So here we can get up to kilobase size deletions. So the model that we started with was to delete the exon, so remove that region of the genome. So now you take away the cell's ability to even make that isoform because the exon important for that isoform simply doesn't exist. So again, gDNA editing can enforce the production of a specific isoform. And now we can ask what happens when we just lose this isoform. So we're calling this technique PigFarm, and it stands for paired guide RNAs for alternative exon removal. And I'll show you how we wanted to adapt this to our studies. So the first thing was we wanted to show that we can use paired guides to delete an exon. We then wanted to use this for high throughput screens. And then we wanted to apply this into an in vivo model, hopefully to study some biologically relevant or clinically relevant systems. So to get PigFarm working, we first looked at an ultra-conserved exon in an RNA binding protein called MBNL. I want to note that this isn't a poison exon. This exon is actually an ultra-conserved exon that contains the nuclear localization signal for MBNL. So just keep that in mind. So we designed a variety of strategies to deliver paired guides to delete this exon. We can use Sanger sequencing to show when we turn on Cas9, we get the expected gDNA deletion. This is an RNA RT-PCR gel where we can show that at time 0, you get about 50-50 ratio of the two isoforms. But at day 7, you get a great reduction in the exon 5 containing isoform. And then most compelling to me, we can do a Western blot for MBNL. And without Cas9, you can see that both MBNL isoforms are produced. But when we use paired guide RNAs to remove that exon, we get specific knockout of the exon 5 containing isoform. And then again, I mentioned that this contains the nuclear localization signal for MBNL. And when we do immunofluorescence, you can see in non-targeting treated cells, and here in orange, MBNL accumulates in the nucleus of the cells, but we can see a loss of MBNL in many of the cells in our paired guide RNA treated cells. Great, so now that we knew the technology worked, and especially that it worked in a polyclonal setting, we can now apply it to high throughput screens. So for this, we can package a variety of paired guide RNAs into lentivirus, add the cells, add the lentivirus to cells, and then let the cells grow over many population doublings. And you then would count the guide RNAs before and after. So the blue guide RNA must have killed the cells, and maybe the red one would help the cells to grow even better. We targeted 500 poison exons, 500 coding exons, and we typically target the 3' splice site of the exons. I'm skipping many details here, but the technology reveals coding exons that are essential for cell viability and also poison exons that are essential for cell viability. Regardless of what cell line we do it in, we often see qualitatively similar results. And we can confirm that for specific substrates, both deleting a coding exon and the poison exon decreases cell viability. And then just in my last 2 slides, I'll just mention, we also got curious in these exons, which seem to have an anti-proliferative effect. But for this, we really switched the model. We added paired guides to cells, put them in a mouse, sequenced them, the guides from the tumors, and found enriched exons. And then when we pick one of those exons, we can show that when we delete that exon, it actually increases cell growth in vivo. So here's the summary. PigFarm is a tool to study isoforms. And I just want to highlight our lab and say thank you to everyone who contributed and our collaborators. And then I'll just put this up while I take questions. Great. Let me turn off. Great, I can hear now. Sorry, I rushed through at the end, but I see there's questions popping up.
Thank you. A very exciting story about poison exons. I'm sure some of you have never even heard about them. So the first question comes from Valentina Snitkova. Wonderful talk, James. Are stop codons more conserved than surrounding nucleotides and poison exons?
Yeah, that's a really good question. I would say that for the classically defined ultra-conserved elements, these are defined as regions where all along a stretch of 200 nucleotides with perfect sequence identity. So in this instance, at least between human, mouse, and rat, it would be perfectly conserved. So we wouldn't see enriched conservation for the 3 nucleotides itself. I think for other highly conserved exons, that might be a possibility, but I haven't looked at that specifically.
Okay, thank you. Then we have a question from Aishwarya Segu. Sorry if I mispronounced your name. Is the ultra-conserved region in the coding region specific to start and stop codons?
Yeah, that's another great question. It's not. So you might remember I showed the ultra conservation around the poison exon of SRSF3, and you can see there that the conservation extends throughout the exon itself, but even into the flanking intronic regions, suggesting that that there's other important regions beyond just the stop codon. One of the sort of models for why that's the case is, again, a lot of these poison exons can have autoregulatory feedback mechanisms. So SRSF3 would bind to its own poison exon to regulate its inclusion. So you might envision that the binding motifs for SRSF3 might need to be conserved in order to maintain the regulation.
Thank you. Then we have a question from Jacob L. Colte. Do you see off-target effects of your pgRNA Cas9 exon skipping method?
Yes, we looked at this a lot, and we looked at it from a variety of ways. So both at the DNA level and the RNA level. So first I'll speak to the DNA level. We did a lot of work using Sanger sequencing of topoclone fragments, I think, for maybe 20 different exons, and we can see that we get the expected on-target effects of the paired guides. So we at least know that we're hitting the right spot. Most importantly for us, though, is the RNA level. And we want to make sure that we're just disrupting the inclusion of that single exon, and we're not just messing up splicing for the entire gene. So you might envision if Cas9 is doing crazy things to the gene structure, you might get the accumulation of all these strange cryptic splice isoforms that would have undesirable functions in the cells. So to address that, we did a lot of RT-PCRs where we took primers and moved them back across the entire message. And we can show that we just get skipping of the intended exon. And we also did a variety of RNA-seq studies to look for low abundance splice junctions. And for all the exons we've looked at currently, it seems to be highly specific.
Do we have time for one more question? Yes. Alison Andros would like to know, do the ultra-conserved regions contain conserved RNA structures?
Yeah, that might be the case. It's not something I've looked at. I mean, I think that there's algorithms available to predict RNA structures. So I think an analysis like that is relatively straightforward and something we can do. I personally haven't looked at it. In terms of structures that I do know about, there's work, and I'm blanking on the group that did this, but there's work showing that ultra-conserved elements are enriched in certain domains of chromatin, or kind of DNA interactions. I don't believe, I can't remember if that study looked at only ultra-conserved enhancer elements or all ultra-conserved regions, but there's also some research showing that these could be enriched in certain DNA elements. So, yeah.
Okay, thank you, James. The time is up. We'll move on to the next speaker, but thanks a lot for the very exciting talk. We now move to Ryoji, who will share with us his insights on transcriptional profiling of specific cell types. Welcome, Ryoji.
Thank you. Let me share my screen. Can people see me? Okay. Okay, thank you, eLife, for giving me this opportunity during these really difficult times. My name's Ryoji, and I'm a postdoc at Harvard Medical School in the CUNY lab. And today I'm very excited to share with you our new method called Probe-Seq, which allows transcriptional profiling of specific cell types from heterogeneous tissue using fish. So, as many of you probably know, you've worked with organs or different tissues, and they're usually composed of many, many different cell types. Depicted here is just one example. It's the retina, which allows us to see. And you can see I'm just depicting the 7 major classes here: the rods, the cones, bipolar cells, horizontal cells, amacrins, RGCs, and Mueller glia. But recent single cell strategies have identified more than 100 different subtypes in the retina. And this is not unique just to the retina. It's also happening in all the other different organs, different tissues, and different organisms. So when you're thinking about doing an RNA-seq experiment to, for example, let's say you want to understand what's going on in one of your cell types of interest during disease, then you have to really think about what kind of experiment you're going to run. So, there are different common transcriptional profiling approaches that are already available. And I'm going to just illustrate this by using a fruit basket as an analogy. So imagine this fruit basket is your favorite tissue or organ that you just dissected. Just like tissue, this fruit basket has different kinds of fruits. And there are some more common fruits like apples here, and some rare ones like kiwis. Now, if you mash all the fruits up and you make a smoothie, then what you're going to do is you're going to be tasting what's the most common fruit, like the apple here, but you're not going to really taste, the kiwis. And this is similar to a whole tissue RNA-seq, in which you get the very common abundant cell types and the transcriptional profiles associated with it, but not really the rare cell types. More recently, we have this fruit salad method, where you can take all the different fruits, basically all of them, and then cut them up, and then you can taste each different ones. But to get to the kiwi, you're going to have to go through all the different apple pieces. And this is something that's like single cell RNA-seq, which is great, and it's very transformative in biology. But for you to get the 1% of the cell type that you're interested in, you might have to go through 99% of unwanted cell types. So in the middle is something where I like to think of it as single fruit bowl. Let's say you're interested in the very rare kiwis, then you can sort them out, you can pick them out, cut them up, and then you can enjoy just the kiwis. And that's something that's like cell type specific RNA sequencing. So there are different transcriptional profiling strategies that are cell type specific already. For example, if you're working with a genetically tractable organism like mice or Drosophila, you might have a promoter or an enhancer for your cell type of interest, and you can drive the expression of a fluorescent protein like GFP and then sort them out. Or if you have an antibody, you're lucky, you'd be able to take them out using antibodies against cell type specific antigens, and again, back sort them. But oftentimes, even if you're working with mice or human or Drosophila, you're not lucky and you don't have these approaches. And it's also a huge problem if you're working with any of these different organisms, these understudied organisms, and antibodies or promoters are just not that available for these organisms. So with Probe-Seq, we had one goal in mind. It was to obtain deep transcriptional profile from any cell type from any organism, potentially. And this is the workflow for Probe-Seq. And as a proof of concept, we decided to go with a fresh mouse retina where we know all the different cell types that are involved. And we decided to take out a very specific subtype of bipolar cells in this layer here. And it's about 2% of all the cells in the retina. So what you do is you can dissociate a fresh mouse retina, and then you can dissociate and then fix in 4% PFA, and you get this fixed single cell suspension. And you can use gene-specific probe sets for your markers of interest. Here we used VSX2 and GRIC1, and VSX2 labels all the different bipolar cells and Mueller glia, and GRIC1, which labels a subset of those cells. And these probes are what we use for Saber fish as well, and you can do this in tissue. And this was developed at least in part by our lab, the Sejal lab. And once you do this overnight incubation with these probes, you can put these green or red fluorescent oligos onto these concatemer tails, and you can use a different combination. And you can FACS sort them and then put them into different tubes. Here we have the VSX2 positive, GRIC1 positive population, and VSX2 positive, GRIC1 negative population, and VSX2 negative population. Then you can decross-link and perform RNA sequencing. Now, if you do this in tissue, this is Saber fish, using the same probes, you can see that VSX2 is confined to the INL layer. These are all the bipolar cells and Mueller glia, and GRIC1, which labels a subset of them. And if you use this on fixed single cells and go through FACS, you can see that there are different peaks. This is the debris peak, and then you can see this is a single cell peak, which is what you want, and other contaminants. And if you take just the single cells, and then you can see this negative population, and there's this bump that is for VSX2. And if you gauge just that, now you can see there's a GRIC1 negative population and a GRIC1 positive population that is very prominent here. And if you look at them under the scope, well, the double negatives, you have the host staining, but you don't have the puncta of VSX2 or GRIC1. Now, the VSX2 positive GRIC1 negative cells, they have the puncta just for VSX2, but not GRIC1, and you have the double positives. And based on the markers we used, GRIC1 and VSX2, we expected bipolar cell subtype 2, 3A, 3B, and 4 to be in the GRIC1 positive population, and other bipolar cells and Mueller glia in the GRIC1 negative, and all other cell types in the VSX2 negative population. If you do RNA sequencing and you look at different markers, which is in these different rows, you can see that in here on the right side are the GRIC1 positive population. you can see that BC2, 3A, 3B, and 4 markers are enriched specifically, but in the GRIC1 negative, all the other markers are enriched. So it seems like Probe-Seq is profiling the correct cell types. And the question we wanted to know was, okay, well, if people are going to use this, how good is Probe-Seq compared to normal bulk RNA sequencing? So this is kind of the gold standard. You have a promoter, we use GRIC1 promoter, driving GFP. So here we electroporated it into the retina. And if you do that, about 72% of GFP positive cells are GRIC1. And then from that retina, from the unlabeled, unelectroporated area, we did Probe-Seq for GRIC1 positive cells and performed RNA sequencing. And in the electroporated area, we fact purified live GRIC1 GFP positive cells using a very standard Trizol approach and performed RNA sequencing. And the results were very good. We thought it was very good. If you compare the transcriptome of the live GRIC1 positive cells and the transcriptome of Probe-Seq GRIC1 positive cells, we saw on an average a correlation of 0.78, indicating that high quality transcriptome can be obtained by Probe-Seq. And if you're interested in not just using 2 probes, but if you're interested in 4 or 5 different probes because you're looking at very specific subtypes, then we thought we can do a serially multiplex Probe-Seq, where we had GAD1, VSX2, GRM6. We had an overnight incubation of all 3 of them, but we put fluorescent probes only on 2 of them, GAD1 and VSX2. And you can see this population of VSX2 positive cells and GAD1 positive cells. And then you can very quickly strip these fluorescent oligonucleotides, but not the probes themselves. And that only takes like 5–10 minutes. And you can see that the labeling is gone. And then you can rehybridize the fluorescent oligonucleotides onto GRM6 here. And you can see a new population appear. So I don't have time to go into this, but we also did this with frozen human retina nuclei, as well as Drosophila midgut and fresh chick retina. And they all worked great. So that shows that maybe this can be possible with any organism. And in the future, I think that Probe-Seq can be combined with a TAC-seq to look at open chromatin in specific cell types, or do single cell RNA sequencing after Probe-Seq, just to get a more finer view of your cell type of interest. And with that, I'd like to thank everybody in the CEPCO lab for being very supportive, especially Connie for being a great mentor, and the Tabin and Demeki labs, biopolymers facility for the sequencing, immunology facts for, and the imaging facility, and the funding source. Thank you.
Thank you very much, Ryoji. Thank you for sharing with us the smoothies, the kiwis, the fruit salad, and particularly the Probe-Seq.
Yeah.
So I would like to start with the first question, which comes over from Hutchinson. Is this method also working for plant cells?
So that's interesting. If there is a way to dissociate plant cells, well, I have no experience with plant cells, but if you can dissociate them, and if you can get these things into the cell wall by permeabilization, I'm not quite sure which permeabilization works for plant cells, but plant cells have RNA, and if in situ works, if in situ hybridization works for plant cells in tissue, then it should work for plant cells.
Okay, the second question is, what are some challenges that you would may encounter if you combine Probe-Seq with single cell RNA sequencing?
Yeah, I get this question a lot. So I think, right now, you have to fix the cells. And if you want to do a high throughput droplet-based method, then all of the decross-linking at the end for Probe-Seq occurs with protease. So if you wanted to combine single cell RNA-seq with the droplet method, then somehow you have to decross-link beforehand and then put them into the droplets. Or you might want to use like a methanol fixation, and that's something that we can try. But I think, at least right now, it's probably already possible to do a plate-based single cell RNA-seq method.
I see. Another question: what are the what is your pipeline for introducing fluorescent nucleotide into the cell? Is it enough to permeabilize the cells after fixing, or do you have some other tricks?
So the probe can go in only once it's fixed and hybridized. So there's a next step, and if it's not permeabilized, I don't know if it can go into the cells.
I see. Then we have a question from Julia Chiesa, who found this talk very cool. What's your pipeline for actually, I think it was the same question, sorry. Can you comment maybe a little bit more about the tricks that you use? Because I mean, you must have been optimizing this a lot.
It's interesting because, so I had a previous paper where we were using antibodies, and basically, once other lab members in our lab got the saber fish to work in tissue, a lot of the same things that they were doing in tissue, if you apply them to single cells, they worked almost perfectly. The only optimization that we had to do was they were using tween in basically all the wash steps. And then I found that if you use tween, the cells clump up a lot. So we just have to get rid of that. But there wasn't that much optimization that had to go into it.
Okay, then we have a question from David Soon. The abundance of these gene markers, is it important? Does the marker has to be very abundant for the method to work?
Yeah, so all of the markers that we used in the paper, they were very abundant genes. The two things that we found that were important were abundance of the genes and the length of the genes, because these probes tile the RNA of interest. So the longer the transcript, you have many more probes that can bind to it. So we found that both of those parameters are important. The good thing is that now that everybody's doing single cell RNA sequencing on basically everything, we have tens to hundreds of different markers that we can use. And these are pretty cheap.
I think, if I understand correctly, we have to wrap up. Is it correct, Naomi? So thank you very much, Ryoji. We now finally move to Sejal. Welcome, Sejal, and thanks, Ryoji. So the last talk will be about this mysterious long abbreviation thingy and its function in astrocytes to regulate sleep homeostasis.
Can you see my screen? Hey, Naomi, is it good?
Yes.
Yeah, great.
It's great.
Okay, perfect. I can see that. Okay, hi everyone. My name is Sejal, and I'm a postdoctoral researcher at the City University in New York. But the work that I'm going to talk to you about today is a work I did as a PhD student in John Van Miles Laboratory at McGill University. So as most of you know, that the most abundant cell types in the mammalian nervous system are astrocytes. So here is a picture of this single astrocyte in the cortex of a rat. As you can appreciate here, that the cells have very profuse, remarkable morphologies. And because of these structures, they are key for neural circuit assembly and functions. So for example, astrocytes, they release different molecules during development that are important for synaptogenesis. because of their morphology, where they sit very close to synapses, they have important functions in neurotransmitter uptake and processing, for example, in expressing neurotransmitter receptors or enzymes. And because of this molecular feature, they can respond to neuronal activity, as well as they release different neuromodulatory factors in response to this neuronal activity. And because of all these key functions that astrocytes serve in the nervous systems, they are very important in different disease mechanisms. So overall, the field is interested in how do astrocytes differentiate and acquire specific molecular profiles? How do they influence CNS function and behavior? And perhaps maybe we can use this knowledge and exploit astrocyte to better understand and develop therapies for human diseases. So I study astrocytes in Drosophila because they have cellular and molecular features that are unserved. And Drosophila, it's a beautiful genetic model system that is easy for in vivo manipulation, and you can readily observe phenotype. So if you were to take a cross section, for example, here it's the larva it's the brain of first instar Drosophila larva. If you were to look at the cross section, you see two histological segments. So here it's called the cortex, which is shown here in green, which harbors all the neuronal cell bodies. And you have this neuropile region that has axons, dendrites, and synapses, but they do not have any neuronal cell bodies. Now, at the intersection of these two histological segments, you have the astrocyte cell bodies that infiltrate that beautiful, membranous branches and connect with synapses and serve in functions. And here is, if you were to label astrocytes in vivo with GFP, that's what it would look like. So again, the question that I'm addressing is, how do astrocytes in Drosophila influence CNS function and behavior? And I'll give you a small example today. That's my PhD work, where I studied this gene called ANUT1 and how it modulates sleep in astrocytes. So before I jump into the results, just want to give you a brief introduction on sleep. That sleep is modulated by two regulatory mechanisms. So if you look here, you have sleep traces of Drosophila, where on the x-axis 0 to 12, it's a light phase, and 12 to 24, it's a dark phase. And you have the sleep profile of individual flies, either in black or in red, and it resembles some of our sleep pattern now that we don't have to go out to work. So this sleep is more or less governed by circadian rhythms, something we call a baseline sleep. However, you can imagine a scenario here that if a fly is sleep deprived overnight, compared to this control fly that is not sleep deprived, the sleep deprived fly exhibits higher amount of sleep the next day, which is something we call a recovery or a rebound sleep. And this is governed by the homeostatic mechanisms. So one of the key questions in the sleep field overall is try to understand the mechanism that governs circadian or the homeostatic rhythms. And there is one example that we know of this mutant, which is in a gene called ANUT1. These mutants, they do not have any defects in their daily sleep baseline rhythms, which is the circadian sleep. However, if you were to look at it in the bottom right corner, you have the wild type flies. When they are sleep deprived in this black bar, they have a higher amount of rebound sleep. The ANUT1 law mutant flies, they exhibit even higher amount of recovery sleep compared to the controls. So how does ANUT1 participate in the sleep functions? So first of all, what this ANUT1 gene is, it's an enzyme called aryl alkylamine N-acetyl transferase that acetylates and inactivates monoamines such as serotonin, dopamine, and octopamine. So the key question is, which cell types in the fly brain express this enzyme? And in order to probe that, I developed an antibody against this exposed region. And we tested this antibody. So what you see here in the control animal, that the antibody beautifully lights up different cells in the nervous system of Drosophila larvae. However, the animal that has a complete deletion of ANAT1 gene does not have any staining, suggesting that this is a ANAT1 specific antibody. So I looked at the ANAT1 positive cells in the Drosophila, adult Drosophila brain. What you see here in the first panel is in green, you have the ANAT1 positive cells, and magenta edges the antibody to reveal the brain morphology. I further did the marker analysis using a pan-neuronal marker LRV and a pan-glial marker called REPO, which suggests that both neurons and glial cells express this enzyme. Based on the morphology of glial cells, I can already pinpoint that these cells are astrocytes, which I further confirmed using a marker analysis where alarm, which is a promoter, which only expresses in astrocytes. So if I were to label astrocytes with nuclear RFP, all astrocytes in the fly brain are positive for ANAT1. And this is sort of a snapshot summary of how we characterized other cell types, where among all glial cells, astrocytes are the key cell type that expresses ANAT1, and there are different subset of neurons that also expresses this enzyme. So here is the model. Until now, we have a very good idea about the circuit that connects a different monoaminergic system and how they generate behavioral output through different receptors. How do astrocytes participate? And here is the one example where I found the expression of ANAT1 in astrocytes. So the question was, do astrocytes alter the level of monoamines via ANAT1 and affect the behavior, in this instance, sleep? And then the second question would be, what monoamines are processed by ANAT1 in vivo? So the first example, the first experiment I did was, what happens if you were to knock down ANAT1 from astrocytes only? And this was achieved with Gal4 system and RNAi. So here is an example. If I were to knock down ANAT1 only from astrocytes, you can only see the neuron-specific expression, but all the astrocytic expression is gone. The number of astrocytes remain unchanged in this manipulation. Here is the sleep data that was done in collaboration with Amita Sejal's lab. So in black and blue, you have the control traces of flies, as I showed earlier. And in green, it's the experimental animals. So what we see is during daytime and nighttime, the amount of sleep remains unchanged. So the circadian sleep component remains unchanged when we knock down ANAT1 from astrocytes. However, in these flies, if we were to do overnight sleep deprivation, as shown here, you can appreciate that the green bars here are the experimental animal, and this is the recovery sleep 6 hours after the sleep deprivation, which is significantly increased in compared to control Gal4 and UAS flies. And this is a very robust phenotype. Now, this phenotype essentially is what was reported before for this ANAT1 low mutant phenotype. So what I'm showing you here is a gene ANAT1 that do not participate in the circadian mechanism of sleep, but it is important for the homeostatic sleep. And just to end here, if I have time, so what happens to the level of monoamines? So when we look at the control versus the ANAT1 low mutant flies, we do not see any difference in serotonin and dopamine levels under baseline sleep conditions. However, if that window is preceded by sleep deprivation, there is a robust increase in both serotonin and the dopamine levels. And if I were to repeat that experiment just by knocking down ANAT1 in astrocytes, we do see a trend in this green bars where there is an increase in serotonin and dopamine. However, it did not reach the level of significance. So just to wrap up here, what I showed you today, we discovered, we found this enzyme A01 that expresses in astrocytes. This enzyme is important to process monoamines. This enzyme is important to drive homeostatic sleep in astrocytes. I did not show you this data, but it is important to drive baseline sleep in neurons. And then this enzyme controls the levels of dopamine and serotonin under sleep deprivation conditions. And with that, I would like to thank my lab and our collaborators, and thank you for your time. And I'll be happy to take your questions.
Thank you very much, Sejal. It was fascinating. I never have been thinking about sleep deprivation and flies in my life, but it's absolutely fascinating. We have the first question from Ariel. How can the understanding of sleep and Drosophila be applied more widely to humans or other vertebrates?
Absolutely, that's a fantastic question. And, just a bit of a historical perspective. So, in 2017, the Nobel Prize went to the discovery of clock genes in Drosophila. And turns out it's the same mechanism that operates in flies that govern circadian rhythms and sleep. It's the same mechanisms that we humans have. So for example, the molecular clock is incredibly conserved. Talking about the A01, which I investigated in Drosophila astrocytes. So A01, it's also present in the human brain, especially in the SCN, which makes melatonin, because this is the enzyme that precedes the first step in the melatonin synthesis. So there's definitely conserved elements. And in terms of circuit mechanism, the monoamine circuit is also very well conserved. So the kind of work we do in Drosophila to understand sleep, it's definitely very informative for human sleep.
Let me see. So Michael actually had a very related question to, are there any human diseases associated with mutations in A01 or any disorders? Has it been looked at genetically?
Absolutely, that's another very nice question. So we looked at the mutation data, and there is actually a syndrome in human, which is in the A01 related gene, which is called the SNAT, that makes melatonin in humans. And these people have something we call delayed sleep onset phase. So they are sort of like, in a way, nocturnal. Their sleep phase runs from late at night, and they have definitely problem waking up early in the morning, so they start their day a little bit late. So that is the one definite example we have where A01 is related to human sleep disorder.
We have a question from Catherine Vogt. Can you see the same effects in a serotonin or dopamine mutant background? Do they have disrupted sleep overall?
Yes. So there's like a laundry list of mutations in genes that are important for serotonin and dopamine pathway. For example, transporters or receptors, and they do exhibit a lot of sleep defects. What was really fascinating from that literature was most of them have baseline sleep deficits, if you think about it, if you already have a baseline sleep defects, and on top of that, if you were to do sleep deprivation experiment, the results are a little bit confounded. So this is the only molecule that had that clean separation, where you do not have a baseline sleep defect, but you only have the homeostatic sleep defect.
I see. I think we have to wrap up. Is that correct, Naomi? Yes, so unfortunately our time is up. We had one more question left, but we have to stop now. So first of all, I would like to thank all these three speakers, Sejal, James, and Ryoji, for sharing their work with us today, which was completely fascinating and very diverse. And I would also like to thank all of you for listening and for asking questions. Our online research talk series will continue next week with 3 neuroscience talks talks on Tuesday at 9 a.m. British time in partnership with Neuromatch Conference. They also have an online unconference. For those of you who don't know what unconference is, it is a conference run bottom up and not top down for computational neuroscience starting on Monday. You can check out their agenda online. Even if neuroscience is not your area of interest, we hope that you will come along to support our fellow early career researchers. And on Thursday, we will be back with talks from Catherine, Megan, and Elka. For updates about this series, please follow us at eLife community, and you can find this schedule and the registration information online. Thank you very much. Thanks for joining, and we hope that you will join again.
Thanks very much for chairing, Anna.
Thank you,
Thank
Anna.
you so much.
Thank you,
Thank you for
everyone.
organizing all this.
Thanks everyone for coming along. Join us on Tuesday.
Yes, absolutely. Have a nice day, everyone.
Thank you very much, Naomi, Miranda, Anya, for organizing this so well. It was fantastic. It was exciting. And thank you very much, all the speakers, for sharing your science and starting this series, which I think is very important in these difficult times.
I agree.
Thank you, everyone. Bye-bye.
Take care.
Bye, everyone.
Bye.
Bye.